Kim. And thanks a lot, Eugenie, for introducing me. And thank you for coming to my talk. As already Eugenie talked about me, before I came here, before I was worked in the UK, my research area was about the animal breeding and genetics for the livestock, for the livestock animal. So today, I will talk about the porcine muscle fiber characteristics to improvement of the both lean meat production ability and meat quality at the same time in the pigs. So the muscle fiber characteristics as considered to the new selection trait, economical trait. And then I was working for to find out the DNA molecular markers, which has responsible for the muscle fiber characteristics, and by use of the candidate gene approaches. So the overview like this, the first part of my talk is about the background. So we will approaching to the answer for the basic questions about what is the animal, or what is the best animal, what is the animal breeding as well. And then I will quickly just an overview about the current pig breeding industries and some of the problem in that area. And then I will talk about my research aims. And next part is about to know about the possibility, to find out the possibility of the muscle fiber characteristics as a new selection trait for the improvement of the lean meat production ability and meat quality as well. So first part is about to talk about the heritabilities of the muscle fiber characteristics, and then the correlations among the other previous economical traits. And then finally, I did some of the cluster association analysis, and I could get some summary of the result. And then to find out the molecular DNA markers, I used the DNA candidate gene approach method. But before I talk about that, I will quickly compare the whole genome approaches versus with the candidate gene approaches. And then to find out the candidate genes, I used the bioinformatical tools for data mining to choose of the candidate gene. And then I will talk about the process of the discovery of the novel DNA markers in a specific gene. So background is about, first question is about what is the animal breeding? I think we are, have various background in this, people. So usually first of all, we thought about the breeding means mating of the selective male with female or some, the other genders to produce of the next generation progeny or some next generation individuals. But the animal breeding, just the general animal breeding, is basically, these kind of the processes. So the most important word in the animal breeding is selection. Selection of the best animal. So what is the best animal? So basically, best animals are estimated, evaluated by the genetic potential of the population or each individuals. So the meaning of the animal breeding is just getting better performance from the current population, current generation to the next generations. So after selection of the best animal, and then to produce of the progenies by specific mating method. And next question is about what is the best animal. So there are lots of livestock or some animals we used, and human beings are using some lots of animals. For example, the dairy cattle. Dairy cattle are usually to produce of the milk. In the past decades, the best dairy cattle is about to produce of the high amount of the milk. And like a beef cattle is a high amount to produce of the high amount of the meat. Some of the horse, racing horse, is fast learning. It's the most important trait, selective trait. In the pigs, there are lots of economical trait, but in the past days, to produce of the lots of progenies, piglets, litter size, like a pet as a cutie or some, as a small size of the pets, or some experimental animal model can be used for the specific purpose, like a disease model or some overexpression transgenic mouse. So for specific, needed some specific breeding method, breeding purpose. But as you can see, these kind of the species, their best animals are totally depend on the species, what we want to make of them. Or sometimes that depend on the time, consumer's mind, in the industry. So nowadays, for example, in the past days, dairy cattle are the most important trait is to amount of the milk size production. But nowadays, the quality of the milk is much better important because of the, some of the lipid content or some amino acid content is much better because of the consumers. Current consumers want to eat, drink some of the high quality of the milk. Samely, I will talk about the current pig breeding industry. Generally, these four kind of the pigs are well used in the porcine breeding area: the Berkshire, Raju White, Yorkshire, Duroc, Landrace as well. In the past decades, the porcine breeders want to increase of their lean meat production abilities, to increase of their red muscle. So as you can see these figures, the past days of the pigs has relatively high percent of the fat, and then compare with the lean meat size. But nowadays, as you can see these figures, high percent of the lean meat size, and then compare the small amount of the fat, because of the Western people who want to eat some lean meat without any fat. But the several problems were produced to only increase of the lean meat production ability. First of all, animal welfare was ignored. For example, every male piglet were castrated by surgical operation without any anesthetics. And some of the piglets were raised in a small pens. And sometimes they ate overfeeding because of the, in a short time, to increase of the average daily gain. And the next part is that they got many of stress from their, ignoring animal welfare. So their disease resistance were significantly decreased, such as, viral disease, PRRS, or PCV2, or foot and mouth disease were increased in the porcine areas. There are big problem in this current porcine industry. And at the same time, the mortality rate, the live litter size were significantly decreased. And today, I will talk about the focus on to the meat quality, because of the meat quality was significantly decreased. But nowadays, current consumers want to eat the high quality of the pork, even though they, paid lots of money, because of you can choose many kind of the meat easily. But the pork is, want to eat some high quality of the pork. But the PSA, the pale softness, exudative pigs has a big problem in the pork quality, or some PSS syndrome, like a porcine stress syndrome, is also based on this kind of the decrease of the meat quality. So these two kind of the characterize of the abilities to big economic trait in the porcine industries. Lean meat production abilities can be explained by two kind of the measured trait. So these two kind of the traits were measured in loin muscle area, loin muscle. So loin eye area measured on the loin size, and the back fat thickness is usually depth of the fat. They can explain about limit production abilities. And meat quality was measured in the loin muscle of the postmortem meat, and the muscle pH, and lightness, and dream loss to check up the water holding capacity. Because of the meat quality can be explained about the postmortal metabolic rate, the speed. So fast metabolic rate means a poor quality, exudative of the water, or some slow metabolic rate means a good, better quality of the port. But very unfortunately, these two kind of the categorized of the abilities has significant negative genetic correlations. That means when the breeders want to increase up the limit production abilities, the next generations, meat quality has poor. So I would like to find some of the bridge trait to increase up the both abilities. That's the reason why I was focused on the muscle fiber characteristics. But usually, muscle fiber characteristics are consisted of their number, total number, or some each type 5 type numbers, and their area after cross-sectional, size of the cross-sectional area, some vertical cut of the muscle fiber. And muscle fiber has several kind of their types, and the big three types are type 1, 2A, 2B. Their composition can be explained about the meat post-mortem meat quality by metabolic rate. So, for example, the black one is called type 1 fiber, is called also red muscle fiber, because of the type 1 fiber has a huge amount of the hemoglobin. But while type 2B fiber has not many hemoglobin, so they call the white muscle fiber. Type 1 fiber has slow contraction time. And the type 2B, in the wild, type 2B, on the other end, type 2B has fast, very fast contraction time. That means they are directly affect to the post-mortem metabolic rate. And they also, type 1 has a high mitochondrial density or high oxidative capacity, and the low glycolytic capacities. And then type 2B, on the other hand, has the opposite characteristics. And the intermediate characteristics to type 2A fiber has intermediate between type 1 and type 2B. So the muscle fiber characteristics can be explained by the number of the total muscle fiber, or each fiber's types or size, and their compositions. So my research aim was about, first one is to know about the possibility to use of the new selection trait for the improvement of the, solve up these unfavorable relationships, the breaking of their unfavorable relationships by use of these kind of the statistical analysis. And then I would like to find some of the DNA molecular markers which is responsible for these muscle fiber characteristics to use of the positional candidate gene approaches. So muscle fiber characteristics has moderate to high heritabilities. That means a high heritability trait can easily, can successful the selection, for the selection. So the next generation, we can get accuracy, increase of the accuracy of the selection. So for example, number of the fiber and the size of the cross-sectional area has moderate and the medium size of the heritabilities, and the fiber types has relatively high heritabilities. So it is easily can be used as a selective trait. But some of the liter size or some of the raw heritability traits cannot be used as a selection trait because of the low heritability is not good to selection. So I just check up the correlations by RAMO programs and among the muscle fiber characteristics and with the other characterized of the economical trait. So red color means a better case to increase or high, or blue color is better case at the decrease or low. When they are low, they are better case. So the size of the muscle fiber has low, low condition can show up the low. That means a better backpack thickness and the better mid-quality conditions, low pH and then high dream loss and then high rightness, like that. And at the same time, high number of the muscle fibers per unit area can show up the better limit production ability, like a backpack thickness, thin backpack thickness, and better mid-quality traits, like a high pH value and then low oxidative poor quality. And at the same time, it is, can show up the fiber type compositions. For example, fiber type 2B fiber area can show up there when they high and when they low, and they show up the better mid quality. That means type 1 fiber is when the type 1 fiber composition is high, or type 2B fiber is low, they can get better mid quality conditions. Like this. So, for example, high number of the muscle fiber, that means a high can get some high number of high type 1 fiber compositions and relatively low type 2B fiber compositions. To summary, I know this is quite a little bit complicated. So now this is a cluster association analysis. Some high group to have which has a high muscle fiber number groups show the some high fiber type 1 and the low fiber type 2 as well. And then they show that the high size of the loin eye area, better limit production abilities, at the same time, better mid quality conditions. So to summarize up this result, the number of the muscle fiber characteristic, muscle fiber number, can affect to the both limit production ability and mid quality at the same time. And especially the composition of the type 1, high fiber type 1 composition and low fiber type 2 composition can affect to directly mid quality conditions. So the criteria of the muscle fiber characteristic we set up to the high number of the muscle fiber and the high composition of the fiber type 1 and the low composition of the fiber type 2 as well. So now I will talk about the candidate gene approaches to which is responsible for the muscle fiber characteristics. Before that, quickly comparing about the whole genome approach versus candidate gene approaches. Nowadays, we can get easily, meet easily high performance data, high performance sequencing data by using of the NGS technology or high density of the SNP chip result because of the much cheaper. But previous years, this is much expensive. So in that time, the candidate gene approach is much useful, the cost effective and time effective. But the whole genome approaches are based on the infinitesimal model. The infinitesimal model is usually, that means the whole genomic area can affect to the specific trait, even though each locus has small, very tiny effect. But the combining of the whole genomic locus of the effect can explain about their specific trait. Like follow up this genomic estimated breeding value. So to use up the whole genome data. And the final purpose of the whole genome approach is to genomic selection, to use up the whole genomic result. And these follow up these steps. But they are still used some of the time consuming or some money consuming because of the use of the population. And they should be genotyping by this high density of the SNP chip analysis. But compared with this whole genome approaches, candidate gene approach is quite fast and then easy to genotyping to use of the only one locus. But it is needed some of the major gene, major effect of the DNA markers. That means that even though small, tiny locus can affect to the specific trait, but one of the big effect, one of the SNP or genomic locus can affect to the big effect to the specific area. That is a quantitative trait loci. And after getting some causative DNA markers, the main purpose is to marker-assisted selection. That means only one of or some several set of the gene or locus to use of them to select of the animal like this. And the other one is culling. Because of this is very quick, because when the piglet or some progenies were produced, and quickly we can check up their genotype by use of the DNA. So, yeah, compared with that, this is but this one of the strong thing is quite, accurate, because to use of the whole genomic data. So now I will talk about the data mining by use of the bioinformatical tools. First of all, first time I use of the GOTOM analysis by use of the human database. And finally, I could get some of the list of the 38 list of the genes annotated in a pig genomes. And then I use up the QTL database. The QTL means a quantitative trait loci. In animal, to get the result from the QTL, that means that they need some of the reference family to the specific trait, like this kind of the abdominal fat percentage or some meat content or ratio of the fat meat, the kind of the traits are needed. So I could get some of the significant regions in a chromosome, chromosome 1, 2, 4, 6, 7, 8 as well. And finally, I could get a list of the locations, which is responsible for these kind of the traits, and the merge of them. And then, to use up the KEGG database, I usually want to focus on each metabolic pathways to muscle fiber formations or lipid metabolisms. The reason why I was focused on the lipid metabolisms, because the muscle can consisted with red muscle and fat. So several the pathways were joined this kind of the formation and metabolisms, but I was focused on to the TGF-beta and Wnt signaling or PPAR gamma, PPAR signaling pathways. And finally, I used up the GWAS result by Porcine 60K SNP chip analysis. But unfortunately, in a Porcine, there are not many of the results for this kind of the GWAS analysis. So I used only back fat thickness or loin muscle area, some of the leg actions. And I could get some of significant genes or locus from the result. And finally, to merge up the whole, these four kind of the categorized of the list of the genes, I could get these four kind of the candidate genes. But today, I will focus on only for PPAR gamma coactivator 1A genes. So PPAR gamma coactivator 1A genes are is also coded by PGC1 alpha gene. And the gene is well known for the can drive of the type 1 muscle fiber formation. As you can see these pictures, and the JUN-D groups in the Michigan University, they made up the transgenic mouse to up regulation of the PGC1 alpha gene. And as you can see this picture, this transgenic mouse has much bigger lead muscle than the wild type. And the C showed up the down-regulated genes. These are down-regulated genes of the PGC1 alpha, and that the down-regulated genes are also up-regulated. And then they checked of the success of the transgenic mouse. And finally, these histochemical figures show that the high transgenic mouse has high % of the type 1 muscle fiber compared with this wild type. So they prove of the transgenic mouse to upregulated of the PGC1 alpha gene can drive up the type 1 muscle fiber formation in a skeletal muscle. And the JUN-D group has also show up the hypothesis, gives some hypothesis of the metabolic pathway joint, including of the PGC1 alpha. When the energy demand demanded, the PGC1 alpha was upregulated, and they can control of the MAP2 and NERF1 as well, some of the downregulated transcriptional factors. And then they can induce their type 1 muscle fibers or oxidative metabolisms in a mitochondria. And the gene has a significant role in various tissues as well. So when the energy was demanded, the PGC1 alpha gene has upregulated, and then they could transform of the specific tissues to like a brown fat or type 1 muscle fiber or high fat, like this, to produce of the high energy. In a porcine, in a pigs, they are mapped. The genes were mapped in a chromosome 8, and to use up their microsatellite markers, they are located in a specific P21 regions. But when I was to find up the DNA markers, I could get some of the sequence data, sequences of the genes, because very unfortunately, until nowadays, the porcine genome sequencing was not finished. So in the times, the building was not good, very poor. So I used together position locus sequence of the gene. So I used the porcine back library, and then after screening, I could get specific clones which has a PGC1 gene sequence. And then fully deep sequencing analysis, I could get full length of the gene sequence. And after population analysis, alignment of the populations, and then I could get some of the novel SNPs in the full length of the genes. And then after find some of the specific method for genotyping by RFLP, restriction, fragments, length polymorphisms. This is a general process for the development of the DNA markers before the association analysis. So first time, I wanted to quickly check of the possibility to useful, of the PGC1 alpha genes for their muscle fiber characteristics. Some of the previous studies were reported about the cDNA sequencing analysis, and then they could discover some of the SNPs. And the gene has 13 exons, and then exon 8 has a relatively long size compared with the other exons. And in exon 8 regions, there is one of the non-synonymous mutations, which has a T to A transversion SNPs. And the mutation can change the amino acid from cysteine to serine. So I just quickly genotyping. No, before that, the other previous studies was studied about the allele distribution of that regions, the locus, and compare with the Chinese pig breed and the Western pig breed. So Chinese pig breed were fixed to the T allele, but while Western pig breed has even distributed T and A allele. So, but I already talked about, most of the pig breeds are used only Western pig breed in these days. So I quickly check up the use of Yorkshire breed, large white breed, and 150 individuals genotyping. After association analysis, I could get this table. And TT group, which has TT genotyping groups, showed up the low size of the small size of the cross-sectional area and high number of the muscle fiber number. At the same time, relatively high fiber number composition compared with the AA groups. And they also showed up the better meat quality conditions. And they're not PSE, pale subcutaneous exudative, some of the problem pork in the TT groups. So it is possible to use to check use up the candidate genes, and I just checked. But as you know about the PPAR gamma coactivator 1A genes, one of the transcription regulation factor. So that means the genes can regulate the many kind of the downregulated genes by the gene regulation. So I just wanted to focus on the regulatory region of the gene. So first time I was focused on the 5' upstream regions to check up the promoter. And the gene has relatively high, like some of the 3 kb of the promoter regions. That means the interesting thing is the gene has no typical transcriptional factor binding sequences, like a Tata box or cat box as well. And at the same time, they're not CPG island in the promoter regions. That means the gene can be controlled by specific many kind of the transcription factors, 6 effect. So I could discover some of the SNPs or some genomic variation, like a microsatellite or some indel. But I was focused on to SNP, which has a restriction fragment, specific restriction fragment size, like this. And after genotyping by RFLP, and then we could get some of the genotypes, 3 types of genotypes on each SNP. But I was focused on only in SNP1, G2A SNPs. The reason why I was focused on G2A SNPs, the SNP was pending to specific transcriptional binding factor site, like as well. The CREB protein was pending to the specific SNPs. And the CREB protein name is the CAMP response element binding. Is a kind of the, which is well known for the transcription factor to increase or decrease of the downregulated of the gene transcription. And then as you can see these figures, the CREB protein has a key role to the regulation of the PGC1 alpha gene to make up the complex with the MAP2 transcriptional factors. And then they could get downregulated gene, control of the downregulated gene, and then as well to increase up the fiber number. So I just checked up the association analysis of the locus, And the GG group has also small size of the muscle fiber area and high number of the muscle fiber, and then high percent of the type 1 fiber, and the relatively low percent of the type 2B fiber as well. And then at the same time, better meat production ability and a better meat quality trace. That means that the locus SNP has significant effect to our potential selective traits. After that, I just quickly check up their role of the transcription expression level by real-time PCR. So GG group has more expressed than compared with the other groups. And to check up the transcriptional activity of this specific allele, G or A, I just make up the construction, plasmid construction, which has a A-specific plasmid or G-specific plasmid. After that, transfection in the mouse CTC12 cell to see the luciferase activities. And they showed that the G allele showed that twice than the A allele. That means, to conclude of this result, the SNP has selectively combining with the CREB protein, and then to make up the selectively combining complex, and they control of the transcription level to the PGC1 alpha genes. And then after finally, they could get effect to the phenotype level. Well, now I will talk about the 3' UTR region of the genes, because nowadays 3' UTR regions has regulated by post-transcriptional regulations by microRNA. The microRNA usually can interact with messenger RNA of the 3' UTR region, and when they make up the interaction complex, they degrade of the messenger RNA, the amount of them. So the interesting thing is the gene, PGC1 alpha gene, has relatively long size of the UTR region. So, as the same with the long size of the promoter regions. That means one of the proof of they can be controlled by some of the downregulated, after post-transcriptional regulation. And their homology also very high with some of the other species. And I also could get some of the genomic variations, microsatellite or index, but I was focused on SNP1 C to T variation. The reason why, also, the SNP was pending for a specific microRNA binding motif. The microRNA 206 has a really well known for the regulate of the myogenic factors. So in the humans, the microRNA 206 are used for the healing of the wounded muscle. So the SNP was selectively, pending for this kind of the microRNA 206 binding motif. So I also check up the phenotype level associations, and the TT group has relatively low muscle fiber type and the high type 1 and high type 2B fiber compositions compared with the C groups. And then TT group also has relatively low quality of the meat quality, pH value. That means, so to check up the messenger RNA expression level, they are not significantly different. But check up the protein level, the TT groups are significantly low protein level, has low protein level compared with the other groups. That means that after expression produce of the messenger RNA by the post-transcriptional level, they are regulated by specific microRNA. So, talk about the implications. The muscle fiber characteristics can be useful for the new selection trait, which can impact to the both limit production ability and milk quality at the same time. And the p-par gamma coactivated the 1A gene can be considered as a major candidate gene for the muscle fiber characteristics. And the gene expression are regulated by the novel SNPs, which can interact with the specific transcription factors or specific microRNAs as well. Thank you for your